This new session about nanomedicine and brain injury in neurological disorder. We have 9 speakers. The speech need to be around 12 minutes with 3 minutes question. And at the end, if we have time, we will have a global discussion. My presentation is the first presentation. So I will start my presentations. First, I would like to thank CleanAm organizer to organize and to continue to organize this meeting in the very difficult COVID-19 context. My presentation will deal with the development of innovative nanotechnology to explore the brain, the inaccessible brain, and also to try to treat it with a nanophysics strategy. Brain disease realized $900 billion medical economic cost because of the failure of most therapeutical trial from neurodegenerative disease to glioblastoma, and this despite preclinical miracle, many disruptive idea and technology. Glioblastoma is probably the worst example with many technology again, and after surgery, most patients, much tumor relapse, and the global survival is only 15 months. So it is a very good paradigm to accelerate the transfer of innovation. The main bottleneck explaining this failure is probably the peritumoral brain inaccessibility. You know, when you do surgery, all the peritumoral brain is invaded by tumor cell. It's impossible to have any information, and it's also very difficult to have therapy that reach this functional area. And in the peritumoral brain, you have tumor cell, but the main driver are electrical activity, inflammatory cell, astrocytes, neuron, and also many physical and chemical modifications such as stiffness and acidosis. We try to develop an innovative strategy using silicon to reach the inaccessible brain. And to the contrary of biopsy, where we cut the tissue, we demonstrate that we can have some kind of molecular and cellular imprint. So, sorry? That you have cellular and molecular
I will take it off.
Imprint on the silicon, and we develop a device, a stylet that enters the brain with a silicon chip. You hide it when you enter the brain, you expose it inside the tissue, and after you do the bioarresting strategy, and you go outside the brain. To do that, it was a very complex process from the cover, from the idea to the translation at the bedside. We have to demonstrate clinical and safety in a non-human primate to develop a specific big animal swine model, to develop the technology to sterilize, and also to implement industrial production inside the clean room of Leti. At the end, we now complete the clinical trial. We include 35 patients. And in this trial, we fully demonstrated the safety, the feasibility to access to the peritumoral area, and the compatibility of the technology with next generation sequencing technology, proteomic and in situ analysis. You can see the MRI of a biopsy inside the brain. You see that you cut the vessel, you also cut the fiber. So the biopsy is something that could be dangerous. And to the opposite, the brain imprint, for example, here when area of highly functional motor area where it is impossible to do a biopsy, you do not cut the vessel and you do not observe any hemorrhage. So now we are, after this trial, moving to next generation technology, adding high resolution bioarresting with nanoporous silicon. This was supported by the Brainomics Nanomed project, associated us to Tel Aviv University and Karolinska. And we had to do again all the process, and it was very long and complex, and we should start a new pilot trial with this new material, that is compatible with single cell analysis and high resolution bioarresting. Similarly, besides the investigation of molecular and cellular even, physics is very important. So we try to add physical properties to the device, optics. You can see here the association of the device with the Monachea stereotaxic confocal strategy. You can see that we enter the brain. It's a human glioblastoma that was labeled by porphyrin, gliolan, and you can see the detection of infiltrative cell as well as you enter inside the infiltrative brain. So this offers the opportunity to enter the brain, to detect the cell, and to catch both the peritumoral brain and specifically to identify the tumor cell. Similarly, and especially in collaboration with Michael Rooks in Caltech, we try to add electrophysiology and also sensor strategy, adding NEMS on the silicon device. In the COVID emergency, we try to develop something that could be useful for COVID using this technology. And as you can see, we had the idea to use a nanoporous silicon to catch not only cell, but low concentrated biomarker in a lateral flow classical assay. We did that with Biotem and MedImprint. We just enter the nanoporous silicon inside the paper. We are now produce and validate the device. You can see the impact for mass spectrometric detection, the crude serum and the nano bioharvesting serum. And you can see that we enhance dramatically the number of low concentrated peptide on the surface on the nanoporous silicon. So we now have a pre-industrial device and we start a clinical trial trying to correlate the signature in saliva and blood to the diagnosis and to the prognostic of COVID. At the end, this technology opened a new rationale for individual precision medicine. First, it offers the opportunity to access to the uncharted peritumoral brain, to access to inaccessible neuropathology complexity, and to move this knowledge by the identification of molecular targets and by the prediction of the connected multi-drug repositioning after devoted bioinformatic study. We move after using this device to investigate the peritoneal area to the need to implement not molecular but physical therapy. Many work, and especially the work of Kim in Nature Material, demonstrates that we can stimulate magnetic particles to induce a mechanical effect that destroys the membrane. With a group of Bernardieni, we develop permalloy nanoparticles, so 60 nanometer to 1.3 micrometer particle, highly sensitive to magnetic stimulation. And you can see here in vitro the dramatic impact of the magnetic stimulation that destroys the cell. So many papers open this potential therapeutic breakthrough. After that, in another RNA nanomedicine, nanofiber, we moved to in vivo application. And it was the first time that people tried to demonstrate the efficacy in vivo in glioblastoma model. And we were very surprised to constate the absence of therapeutical effect. And when we check the penetration of the particles, we realized that the particle did not penetrate the brain and that they remain outside outside the cell in the extracellular space. So completely to the opposite of the original hypothesis. So we move to a next generation in vitro model, not bidimensional model, but three-dimensional organoid glioblastoma model. And we use this model, as you can see here, to inject the particle, and we stimulate them, and we confirm that in this condition, we do not enter the cell, and we did not observe any proliferation effect. But in this much more relevant model, we demonstrate, as you can see here, you can see a control with proliferation, but also cells that migrate inside the gel. And you can see that at 20 Hertz stimulation, we completely neutralize the migration and invasion of the cell. You can see that at higher magnification in the control, and the mechanical stimulation completely neutralize the lamellipedium that enters the brain. So we completely move the paradigm from a local cell nanomechanical description to a new concept of remote anti-invasive effect that could be very easy to apply in vivo in the human model. In conclusion, we develop a new biomedical procedure, nanomolecular and cellular high-resolution imprint. We did the clinical validation and for the safety and operability, and now we move to Tyrannostic Trial. It will be very important to have physics to the molecular exploration. We just show you an example of what we can do. And also for the prevention, to use this technology to explore the environmental contamination inside the brain. That is a very important international concern. For that, one very important bottleneck will be the need for innovative smart data strategy, and we really need new tools. And to conclude, you know, with the COVID crisis, we had many miracle technology, therapy, and lab on chip. At the bedside, we have nothing. And I think that COVID is a very good example that beside the cover in high impact factor review, we also need to support translation that is slow, expensive, but that is very important. Thank you for your attention, and I would like to ask all the European partners in these projects. Thank you very much. So do you have any questions? So I check. So if you, no, I just check if there is online question. No question. So we have in time. So I think we can move to the next presentation done by Ruth Gabizon from the Department of Neurology, Hadassah University Hospital in Jerusalem, nano-formulation of pomegranate seed oil. So thank you very much, Ruth. You can start your presentation.
Here I'm mute, and I'm going to load my presentation in a sec. Share screen. Yes. Is it okay? Can you see this? Hello? Okay.
It's perfect.
It's perfect. Okay. Then I'll start. Yes. I'm going to tell you today, first, thank you for being here and for inviting me to this meeting. And I'm really sorry we cannot do this in Basel, which I'm sure everybody will enjoy, but that's life these days. So I'm going to tell you about Granagard, our food supplement, which is a nano-formulation of pomegranate seed oil that we have developed for the prevention of neurodegenerative diseases. So the first thing to discuss with you is what is a smart food supplement and how we define this. So these are, we see it as reagents that maximize the biological activity of natural ingredients. They increase bioavailability, dose, and distribution to target organs. However, they are tested for their biological activity and mechanism by the most rigorous scientific criteria, as if they were drugs. They are safe for long-term administration to the general public, and their ingredients are designated as GRAS or generally regarded as safe as designated by the FDA. Well, I'm going to tell you more actually about the first thing that we have developed is about neurodegeneration, because this was actually my subject of research for my life. So neurodegeneration is the umbrella term for the progressive loss of neurons. And among neurodegenerative disease, we include prion diseases, Parkinson, Alzheimer, Huntington, as well as ALS and even late stages of multiple sclerosis. And as the population is aging, neurodegenerative conditions such as Alzheimer become actually our next great crusade. Of course, this was before we even believed that just a virus can change our life. So what are the main pathological features of neurodegenerative diseases? So there is aggregation of key disease proteins such as A-beta for AD, synuclein for Parkinson, PRP for CJD or other prion diseases. Each of those has one or even more of these proteins that change their conformation and aggregate. These are aberrant aggregated proteins. They cause oxidative stress, overreaction of reactive oxygen species, and elevated ROS levels cause mitochondrial dysfunction and cell death. And this is how we can actually start from an aggregated protein that can happen just by chance or genetic because of a mutation in these proteins, and eventually neurons die, and we have a neurodegenerative disease. So what are the treatment options for this kind of diseases as a whole? First, we have the, obviously thinking about dismantling or inhibition of formation of a specific key protein aggregates. And this could be antibodies, small molecules, reduced expression by all kinds of RNA interference reagents. But we know that this is very expensive, and the estimated cost for an antibody medication can get to $200,000 or something dollars per patient, and this is very difficult to come by. And actually, again, going back to our situation here with COVID, we are more now have to think about a treatment that will be for the whole population, and we cannot actually, will not be able to spend that much money for one patient per treatment, I guess, in the future. For the all conditions, we can think about neuroprotection, reduction of prevention of neuronal death by simpler pathways and the oxidative damage and metabolic abnormalities. So we can see this as a way of delaying the disease or prevention. And obviously there is the cocktail of both options in the future that we can see as a general treatment. But now let's a little bit go back and think about even if it's even possible to cure or reverse the damage in neurodegenerative diseases? Well, we are used to regular diseases outside the CNS, there are clinical symptoms, then there is diagnosis, treatment, hopefully, not always, but hopefully in recovery, then you go back to normal life. This is the way we are used to see symptoms and disease. However, in the brain, this is totally different because usually the clinical symptoms are late and they appear when the neurons are already damaged from the beginning. So when we first see the symptoms, there is no going back to normal life, even if the best treatment was available at this point. Neurons are already dead and we cannot revive them. So we have to think about another form of thinking about this and what can we do? So we think we should do preventive treatment for at-risk individuals so we can start from the beginning and not after it's too late. So what are the obligatory properties for preventive treatments? I mean, how can we even think about this? So they should be safe for long-term administration to healthy individuals because we are now going to treat healthy individuals and not very sick individuals. So it's a totally different condition. They should be compatible with most all maintenance drugs, meaning medications prescribed to treat common chronic conditions as the high blood pressure, heart disease, diabetes, high cholesterol, and others. They should be compatible with those and not interfere with each other. The price should be reasonable and affordable. Most of the aging population will require neuroprotection at some point, so we cannot, as I go back to the antibodies, go back to $100,000 a year or more for each of the patients. And based on these criteria, we decided to produce a smart food supplement with targeted neuroprotective activity. So this is our food supplement. We call it GranaGuard, and it's a nano droplet formulation of pomegranate seed oil. And why this? PSO, this oil contains high concentrations between 80 to 90 of punicic acid, also known as omega-5, which is a polyunsaturated fatty acid with 3 conjugated double bonds, considered one of the strongest natural antioxidants. It's only present actually in pomegranate seed oil and in a Chinese plant where there is only 40% of it, so it's totally specific. PA metabolizes into one of the forms of conjugated linoleic acid, CLA, both an antioxidant and a calpain inhibitor, and shown also to inhibit A-beta oligomerization in Alzheimer's disease cellular models. So if we can just bring it to the brain, then maybe we can use this as a preventive treatment. And indeed, we see there is an increased bioavailability of punicic acid if we just take the oil and convert it into nanodroplets by specific surfactants. If we do this, then and measure how much punicic acid or CLA we can find in the liver and in the brain, we see that when we administer to mouse the nano-PSO formulation instead of the regular PSO formulation, we find much more CLA. The metabolism from PSO to CLA is increased significantly, and this is when we find it in the brain. We cannot find punicic acid at all, but CLA in the brain is only present when we treat these mice for weeks or even for months with our nano-formulation, and now we have a significant amount of it in the brain. Our results show, therefore, that small mice cells may facilitate the conversion of PSO to CLA in the liver and subsequently the targeting of CLA into the brain. So we have tried to see what this reagent can do to different models, animal models of neurological disease. This is our transgenic mice for genetic Creutzfeldt-Jakob disease. These mice are born healthy, and about 6 to 6 months, they start to show neurological symptoms, and they died between 1 year to 14 months. There is the Alzheimer 5-FAD model, which is also actually a genetic model of Alzheimer's disease, and these mice between 5 and 10 months show deficit in memory. And this is EAE, the model of multiple sclerosis, which is also at old age a neurodegenerative disease. We have tried it on brain injury and of models of Parkinson as well. So what is the effect of Granagard on these animal models of brain disease? In the TG mice for genetic CJD, which for us in Israel is an important model since Libyan Jews have this mutation, and actually most of the genetic CJD patients in Israel are of Libyan origin with this mutation. Administration of Granagard to these mice, they prevent and delay disease aggravation for 6 months, something totally unseen in this disease. For the 5FAD mice, the model of Alzheimer's disease, administration of Granagard protected memory and reduced neuronal death. For EAE, the model of multiple sclerosis, administration of Granagard resulted in significant reduction of disease burden, reduced of both the myelination and oxidation of lipids in the brain. And in the brain injury model, administration of Granagard reduced memory loss and neuronal death. So this is what one of the experiments we've done with the Alzheimer model. This is day 1. You let the mice go only on one side of the maze, and on day 2, you open again the other side, and you can see it. Normal mice on day 2 have been here, so they will prefer to go on the other arm. However, the AD mice, which they don't remember they've been here, and they will go the same for both arms of the maze. And indeed, when we see this, this is exactly what we can see, the untreated TGs, the treated ones, and even actually, surprisingly, this is also true for the control wild-type mice and the treated wild-type mice. So we see some kind of increased memory in the transgenic mice and also in wild-type mice. When we look what happened to the A-beta levels, the aggregated protein specific for Alzheimer's disease, and this is the 5FAD mice at different ages. We see some aggregation and a lot of aggregation at 10 months of age. The treated mice that were treated from 3 months of age, we see extracellular beta levels decreased in the Granagard treated groups. And if we just look at cells in the brain themselves, we see that the green is for the A-beta aggregates. We see much less A-beta inside the cells, and actually no A-beta more in the mitochondria. Since the COX-4-1 and mitochondrial protein, now we can see it in red without any A-beta, and here we can see it in orange together with A-beta. So Granagard administration resulted in a significant decrease in the aggregation of A-beta levels. The mitochondrial A-beta does not accumulate under Granagard treatment. So this is, as I said, the patented self-emulsion formulation of nano-PSO, the nano-formulation of pomegranate seed oils. There is a special mixing formulation for it. Then when we take a drop of water, a drop of the mixture into water, we see the emulsion. It's usually for a long time with the same size between 200 and 300 nanometers. And we have tested it in many animal models, encapsulated into soft gel capsules. And this is actually what happens in the stomach. And this is our product. Now, the vision of Granulix, our startup biotechnology company, is to put into the market a sophisticated food supplement with strong neuroprotective properties. Granagard, as I told you, is a brain-targeted nanodroplet formulation of pomegranate seed oil, shown to be effective in different animal models. We are now testing Granagard in several clinical trials, MCI, which is the first steps of Alzheimer, and actually we are also starting a clinical trial on post-COVID patients with complaints of cognitive reduction and memory loss. We have already done an experiment in MS patients and demonstrated significant memory improvement. And in the future, now we are producing new products on this kind of same platform. And these are all the people that work with me in my lab. Professor Shlomo Magdassi, our nanotechnology expert, head of the nano unit in the Hebrew University, doctors from different places in Israel. And thank you very much for your attention.
Thank you very much for this excellent presentation. Any question in the speaker? I check with no internet question. So maybe just one. What is your strategy? Do you want to develop a nutritional product or a pharmaceutical product that will need clinical trial, for example, to demonstrate that you have an impact on Alzheimer's disease or the neurodegeneration?
Well, and our strategy is we already have this product into the market for several years. And because it's a safe food supplement, regulatory, you can sell it, and then you can at the same time do clinical trials, and we do several. We have done an MS trial, we are doing MCI tests in Rambam Hospital, we are going to do the COVID experiment in Hadassah, and we actually have a huge number of families with a mutation of the Libyan Jew mutation for Creutzfeldt-Jakob disease, and we are following these individuals for several years now to see if they will get sick with this mutation. Usually disease appears between 50 to 60 years of old, the majority of people. Until now, none of those have become sick, and actually we believe there are less patients in the last few years in Israel. So this is not enough statistically, but it's encouraging for now.
Okay, thank you very much. Well, I think we'll now move to the next speaker, Mira Marquez-Kalish from International Research Affairs in Tel Aviv University. The presentation is Brain Clinical Informatics, Promise and Barrier Towards Precise Medicine. Thank you very much, Mira. You can start. We don't have sound. Mira, oui. Microphone. Good.
Can you hear me now?
It's perfect.
Okay. So good afternoon from Tel Aviv. Thank you very much, François. And many thanks to Biat and Patrick and Joshua and the technician that helped me right now. So I hope it will work, but I do hope that next year we will meet all together, like in the old days in Basel, and we will talk and smile and look and share. And, that's what I really hope. But meanwhile, I think we need to learn a lot of lessons from this pandemic. And there is a lot to learn. The first of it is to look on the broad picture, like we see here Monet, and we need, now it doesn't move. So what shall I do? Okay, we'll see. I hope it will work technically. So I move to the next slide. So what I said is what the pandemic taught us is that the world is one. No borders, no continents, no countries. The pandemic is everywhere. Therefore, we should look on the broad picture and of the human body or the human being and its surrounding as one functioning system. And that means to look on the micro, on the in-body, and to look on the macro, on the mental and physical environment, and to look on the broad band of understanding and knowledge and science in order to define a real precise disease signature. And, converging all knowledge and data and whatever we can. And as you can see here, down the various technologies that we call them converging technology. And that brings me back to the old day when we started, with the NSF and then with Europe, talking about converging technology, talking about the third revolution by Phil Sharp and Alan Leshner, and this special book that came up in joint venture that I had the privilege to be part of. But the thing is that converging knowledge and technology, and I would say for society. And the big barrier is, could we really provide precise disease signature, or as we said, personalized disease signature? I think we need to try very hard, but we can't really still provide it. So therefore, what I want to focus in this very short lecture is on brain disease signature. Following the two previous talks that I had the privilege to be in their session. So I want to look on here on all what we were able to identify in the various science research and implementation, all the data and knowledge that we can combine regarding the brain and the body, and build the bridge in a very reliable, upon regulation, keeping ethics and privacy towards providing a real, I don't know if you can see it all, quality of life. The best quality of life, and especially with the pandemic, and especially these days, that the vulnerable group are mainly the elderly and the kids, given all the loneliness and all those separations and so on. So I think this is a very big goal, especially nowadays. And I will relate to the Human Brain Project as I'm part of, and mainly to the medical informatics. And when we are talking about the medical informatics, we were trying to combine and build a platform, bringing on board various hospitals across countries, across cultures, across clinical protocols or local regulation, and see whether we can combine the data again for the benefit of looking at the big picture and providing the best brain disease signature. Nowadays, we are doing it. We have started. We have 30 hospitals on board, not with full data. We are doing it federated. It's not what I'm dreaming, but it's on the way. And the reason I'm bringing all that and saying that we have to do precise and reliable is just to give you one example. And Ruth talked about Alzheimer's. So one of the best cohort of Alzheimer is ADNI, the ADNI American Data Bank. But when we looked on that few years ago, we saw that the best predictor is the missing values. And you can hardly predict 84%. So it's really not the way we need to be as reliable as we are promising, especially for personalized and precise. So we developed the tools in order to build a curated data with full records. And I won't go into all the technology, but luckily enough, and that to give you a hope, we were able to use different, and that's the way to use different schemes and different statistical tools and technologies. The WISBA is rule discovery, the other one is clustering, and we were able to really identify subtypes, and I will skip that. That's another heat map line that we were looking on different brain regions, but I won't go into that. But the idea is to use all technology, to use the best statistical tools, and really to go as near as we can to reliable, replicable outcome. So we developed a new scheme that was a new methodology that was starting with utilizing the knowledge of the professionals, of the doctors, because they have the experience and skills, and then move what we call to clusters analysis, looking on the data, analyzing it without pre-assumption, and then to add the biomarkers and all the other data, visualization and so on, MRI. And then what we did, I wouldn't go because I showed it already. I wouldn't go further into that, but I want to show you, I don't know if you see it, what's running now. So we took the data, and at the bottom, you can see the assignment of the ADNI data bank. And on the top, the clusters that we identified very well. And this gives a hope. And luckily enough, we were able to identify 2 new subtypes that weren't known. And as you can see, we went further and we verified those 2 subtypes with the Gemini Italian 14 labs. And you can see both of them. And then we published, so if I'm talking too fast and I'm jumping, there is a paper that you can see and verify. But this is the hope, and therefore we can do better, and we can really reach out to precise and personalized medicine. But what I want to focus in this very short lecture is about the PTSD, the post-traumatic symptom disorder, that the corona brought it. And just, you know, there was an announcement that more and more people approach the hospital complaining about PTSD. It was announced in Israel. I'm sure it's the same all over the world. And as I said, I'm sure it's the same for the elderly and for the kids. And so we started from the day one that the patient were approaching us, and it doesn't matter whether it was avoidance or whether it was negative emotion, or hyper arousal, whatever was there, we started with questionnaire, and we started with the telephone calls, and with the physician, and with the psychiatrist, and we collected the data, the clinical assessment, the cognitive functional measures, and we had our very good people, both at the hospital and, using the software and so on, and then moving to MRI and imaging and combining all this big data. And the idea was to look on day one when we meet them, and then after a month, and then after 6 months, and then after 14 months. And it was very blessed PhD research of Ziv Ben-Zion that you saw. And again, we took this data, this big merge of data, and we applied our 3C method. And we started again from the physicians from their knowledge and experience, and then moved to supervised analysis of the whole data, and then added the biomarkers and did the unsupervised analysis. And we were lucky enough to be successful with that, another paper. And as you can see, we identified the 2 subtypes, the PTSD and the non-PTSD. Just using this, and it looks very, I hope you see it because I see the picture of the audience here on the slide, so I'm not sure. Can you see it clearly, the 2 subtypes? Okay. And then we took those subtypes, and to identify a specific individual, we used the decision trees. So those are just, the first subtypes that we did. And then we wanted to do an early classification and prediction on as soon as we can. And we were looking on other measurements to be used for our prediction, like cognitive flexibility and low hippocampal volume. And also what we can use today so much easier is the MRI, and to try to help those more and more patients, and as I said, especially the elderly, as early as we can. And then I want to give you some hope again. regarding the pandemic and nowadays, that when we were looking, just as an anecdote on this PTSD versus no PTSD people, the PTSD patients were less willing to take risks. They were more, they took better care of themselves. And while talking about the pandemic, I think it's very relevant. If they will wear the masks and everything and take care, maybe they will be better off. So, it's not always the bad symptoms. So just to give you one anecdote as an outcome. And there I want to come back, and Ruth talked about that, and we heard it yesterday in many lectures and today as well, about the healthy aging, about, the things that some say we left alone, those care homes and those aging people, especially nowadays, some people say even worse, that we care more about the young than about the elderly. And thus we established a healthy aging international initiative at Tel Aviv University. You can go into this link and listen to all the lecture in the two first symposiums that we had in Tel Aviv. And more than that, last week we had a session, a joint healthy aging symposium with BIRACS, the bilateral Israel-UK, and Sarah Harper said that this is our duty of today. Everyone, especially in the pandemic, not to live alone those healthy aging, loneliness, and to take better care of themselves, of this. With that, I want to thank you with this message, very important message to all of us. Thank you very much. We can't hear you.
Thank you very much, Mira, for this very nice presentation. Any question? No?
If there are not questions, I have a small aperitif for you about healthy aging. So but check first if there are questions.
Okay.
Okay, so I will do it. And I just want to tell you that the best place in the world is just a nice tour, like Biat is doing in the breaks. This is the Dead Sea, but it's one of the best places for healthy aging. And the reason is that it's the sun is safe. We have 6% more partial oxygen. We have all the mud and all this salty water. And these are the diseases that can be helped just by being at the Dead Sea. And most importantly, nowadays, especially with the pandemic, is the cardiovascular disease that we have proven results because of the 6% more partial oxygen and many other things, and non-polluted, non-allergenic, which is also correlated with the pandemic, and all those diseases. So just to give you, once you can fly, you're mostly welcome. We established a Dead Sea Research Institute there for the benefit of all. Thank you.
Thank you, Mira. We will go for confinement in the Dead Sea. So no more question. I think just one. I think this is kind of stratification, smart data stratification is very, very important, and especially for clinical trial. Do you have any example where this was introduced in the neuroprotective trial, for example, with a monoclonal antibody in Alzheimer's disease or something else?
The examples that we have with this 3C and other technique that we were using, we were working, as I said, on Alzheimer's disease. We were working on Parkinson, and everything is published, so you can find that. And we were working on the PTSD. And nowadays we are going in 3 other, but we didn't reach yet clinical trials.
Okay, thank you very much, Mira. So we are now moving to the next presentation done by Professor Bae-Ing Hong from the Department of Chemistry in Seoul National University. The title is Graphene Content Dot for Parkinson's Disease. Thank you. We can start.
Hello?
Perfect.
Okay, thank you very much for the introduction. And then I'd like to introduce our recent results on graphene quantum dots for neurodegenerative disease. So as you all know, native alpha-synuclein is dispersed in our brain. There's no problem. But for some reason, if some misfolding happens, it aggregates into oligomers and then amyloid fibrils, and finally kills the dopamine neuron. And people are trying to develop many the therapy to block this this fibrillation of alpha-synuclein. And then we actually tried to use this Congo red, and then we found that it shows significant toxicity, and it's very hard to deliver through the BBB layers. In general, the amphiphilic molecules are capable of dissolving the fibers, but problem is that they cannot penetrate BBB layers. The graphene is a single layer of graphite, and then graphene quantum dot is the smallest particle of graphene, and then is basal plane. Central area is hydrophobic, and then edge is hydrophilic. So probably you can think of kind of two-dimensional surfactant. And then advantage is that you can attach any molecules or through the covalent bonds or non-covalent bond. So graphene oxide or graphene quantum dots can be a good DDS system. And then we have used the typical animal model of using alpha-synuclein PFFs. So we sonicate the PFF fibrils and to make the oligomers or the monomer particles, and then inject it into the brain, and then we can induce the symptom of the Parkinson's disease. And then what we found is that during this incubation of alpha-synuclein monomers to form fibrils, if you add graphene quantum dots, you can almost completely block the formation of alpha-synuclein fibrils. This can be confirmed by THD fluorescence or turbidity assays, as you see here. And then the surprising thing was that even for preformed alpha-synuclein fibrils, if you add graphene quantum dots into this solution, then within 7 days, alpha-synuclein fibers can be completely dissolved like this. And then in this BN page analysis, we found that the polymeric, the fibrils of the alpha-synucleins dissolved into alpha-synuclein, the monomers. The important thing is that the alpha-synuclein monomer is toxic or non-toxic. And then, so in addition, we found that graphene quantum dots inhibited the fibrillation process in primarily cultured neurons and prevented PFFs induced neuron death. So as you see, compared to the PFF case, when it is treated with graphene quantum dots, you can suppress the neuron death. And also we found that the mitochondrial dysfunction can be also prevents the mitochondrial dysfunction mediated to abnormal cellular reparation and oxidative stress because graphene quantum dot is capable of scavenging, kind of reactive oxygen species because of the very unique electrical property of graphene. So any radicals or unstable states can be stabilized by graphene. And also we found that graphene quantum dot inhibits neuron-to-neuron transmission of seeded alpha-synuclein PFFs, so confirmed by these microfluidic chambers. So if you add graphene quantum dot into the first chamber or second chamber, you can block the transmission into the neighboring chamber. And this is the in vivo result after sacrificing the mouse, and then we check the density of fibrils inside in the brain. Graphene quantum that treated the case shows the less, formation of the fibrils. And this is the so-called cylinder test. Compared to the graphene quantum dot treated case, if it is not treated, the graphene quantum dots, actually we injected the graphene quantum dots every 2 weeks for 6 months, and then found that in the control case, the mouse can use only less than 30% of 4-pole, but the graphene treated one is close to normal level. And also we checked the long-term evaluation of the toxicity and found that there's no accumulation in liver, the kidney or spleen. And then, so it can be exerted by urine like this. So the half-life is about 2 weeks. So that there's no long-term accumulation, and then graphene quantum dots can be removed almost 50% in 2 weeks, so that we are sure that graphene is not quantum dot is not toxic. And this is simulation study showing the dissolving process of the backbones of alpha-synuclein fibrils by graphene quantum dots. So as you see here, because of the amphiphilic kind of surfactant-like property of graphene quantum dot, it dissolves the backbone of composed of beta sheets. And if you look at the CD spectra, and then as you know, we can calculate the percent of the alpha-synuclein or alpha helix or beta sheet components, and then found that initially the beta sheets are rich in alpha-synuclein fibrils, but after treatment with graphene quantum dot, it comes back to the normal level, and then you see a huge increase in alpha helix contents. This means that graphene quantum dot dissolves the alpha-synuclein fibrils, and then the final product is native alpha-synuclein monomer, non-toxic monomer. And we also see some simulated study for the in vitro BBB study, and then found that the graphene quantum dot is very selective. And then if it's a little bit larger, the size is usually 2 to 3 nanometer, but if it is 10 times larger, it cannot penetrate BBB layers. And graphene quantum dot is a little bit negatively charged, but if you change the charge, it cannot penetrate the BBB layer. And here we can label the graphene quantum dot with biotin, and then measure the existence of graphene quantum dot using the fluorescence. And they found that they are going to the lysosome first, and then exerted from the body. And we compare that IP and IV injection, and there's almost no difference between them. And as you see here, this is a typical IR spectrum of graphene quantum dots. And then if you use a little bit larger graphene oxide, or if you remove some oxygen species by reducing agent, actually those are not effective as a therapy. So we now confirm that the size is important, and also functionality is also important. And then we try to analyze or interpret the mechanism of the graphene quantum dot. So I'm a physical chemist. I'm not a biologist, and then as a chemist, probably we can regard our body system as a kind of the reactor in a beaker. So there is always the equilibrium between enthalpy and entropy, and then the the reaction direction is decided by the free energy. If free energy is negative, then the forward reaction is favored. But if it's positive, the reverse reaction is favorable. So in terms of entropy, so there are a lot of random particles, random monoparticles are dissolved in our brain, but they are forming oligomer and then the single particle. That this is really unfavorable in terms of the entropy. Entropy actually loves the the randomness, but because of some gain in enthalpy, this forward reaction is favored. But if you increase the randomness factor, if you maximize the entropy factor, actually you can reverse the reaction like this. So people think that the formation of alpha-synuclein fibril is irreversible, but actually in chemistry there's no irreversible reaction. So very slow reverse reaction is possible always, but by introducing graphene quantum dots, you can maximize the entropy factor, and then you can induce the reverse reaction, so that you can dissolve these fibers into oligomers and oligomers into monomers, native monomers. This is very different, therapy mechanism, mode of action, different from the previously suggested therapy for CNS disease. So this is case of amyloid beta. We confirm the same phenomena for the A beta fibrils within a week. All the fibers are dissolved by graphene quantum dots. Even for tau fibers, they are dissolved into the monomers. And then we also recently finished the in vivo test for the simulation test, and then this shows that why this backbone of the fibers are dissolved by graphene quantum dots. So graphene, there's a lot of hydrophobic phase, and then the interaction between the hydrophobic phase and then the beta sheets are very so strong, so that the backbones can be destroyed by the graphene quantum dots. This is our recent in vivo results. After 8 months of the 5x FAD transgenic mice, so we inject graphene quantum dots 3 times, the bi-weekly, and then there's a dramatic change. So this is control, and then after injection, the density of alpha beta aggregates has been reduced more than 60%, 50%, so that its graphene quantum dot treatment is very effective. It can deliver into our brain, the brain of mice, through the BBB layers. It dissolves the fibrillation. Then we also found that the therapeutic effects of graphene quantum dots for the Alzheimer model. Okay, so graphene quantum dots are, they are identical, the single therapeutic particles, and then it can be extended into different pipeline, including Niemann-Pick disease, Crohn's disease, and liver disease. All those are related to inflammation or fibrosis and so on. So this unique property of graphene quantum dot dissolving fibers, and the ROS scavenging properties, the BBB penetrating properties, those are very advantageous for various pipelines. So most of the research has been done in collaboration with Johns Hopkins and the Seoul National University, and I especially thank Professor Han-seok Go in Johns Hopkins University and my students, Jaemin Yu and Dr. Dong Kim. Thank you for your attention.
Thank you very much for this impressive presentation. Any question from the previous speaker? No? So maybe I have one. I was very impressed by the potential therapeutical application, but one concern with, for example, carbon nanotubes or graphene is the microglial reactivity inside the brain that we know to be activated, for example, in the preclinical model that you use. So did you investigate the microglial reactivity and the potential secretion of cytokine when you contact, for example, macrophage with your graphene? Because I think it's very important for clinical application.
Yeah, yes, we did the microglial activation. Probably, I have some slides for that. So you are asking, microglial activation, right?
Yes. Okay.
So,
very interesting.
So anyway, we don't know anyway, the inflammation can be, effectively suppressed by graphene quantum dots. And then as you see here, so microglial activation can be suppressed by the graphene quantum dots. This is different motor, Niemann peak type C motor, but it's mostly the same for the CNS disease. And also we analyze cytokines and the level. So probably we have also additional mechanism that suppresses inflammation for CNS disease.
Okay. Thank you very much. No more questions. So I think we can move to the next presentation done by Dr. Zara Almadi from School of Science and Technology in Nottingham. The title is Selective Entry of Lipid Vesicle into the Brain Post-Intracerebral Hemorrhage Offers Novel Therapeutic Opportunities.
Thank you very much for the introduction, and I would like also to thank the organizers for making this CleanAm virtual conference to happen, and hopefully we can meet all together again in the coming years. So today I would like to share with you some of the work that we did on using lipid nanoparticles to selectively target the hemorrhagic region in intracerebral hemorrhage. So intracerebral hemorrhage occur when a diseased blood vessels ruptures, and this allows the blood to leak into the surrounding brain. It happens or it contributes to about 10 to 15% of all the stroke cases. And there are different causes for this disease, which could either happen due to vascular remodeling or malformation, but could also happen secondary to other conditions such as trauma, brain tumor, or even the use of anticoagulant medications. However, disregard the cause of intracerebral hemorrhage is usually resulted in either primary brain injury, and this is as a result of the accumulation of a blood product within the brain, but also resulted in a secondary brain damage that's caused by the physiological and the pathological response to the hematoma. So although intracerebral hemorrhage occurs in only a fraction of the stroke patients, the mortality and the quality of the life for the survivals is very bad, and therefore it represents a significant public health burden. So in terms of treatment that is available for intracerebral hemorrhage at the moment is all supportive treatment that try to maintain the neuronal function, mainly by reducing the intracranial pressure for these patients. However, until now, we don't have any treatment that can prevent any damage to the neuronal tissues or promote their recovery. And one of the main restrictions for getting treatment for this disease is the protective nature of the blood-brain barrier that limits the delivery of many of the therapeutic molecules into the brain. However, we know now from evidence from both preclinical studies and also from clinical data that the blood-brain barrier integrity is altered in intracerebral hemorrhage. So we think that this could provide a window of opportunity in order to get some drugs into the brain. So therefore, understanding the mechanism and the time frame of this blood-brain barrier alteration that is occurring in ICH is very crucial for the development of potential treatments. So in my lab, we are very much interested in understanding the changes in the blood-brain barrier alterations in different neurological conditions. And recently, we have demonstrated that we can take advantage of a blood-brain barrier disruption in ischemic stroke to translocate liposomes into the brain. And we have shown that this accumulation is very specific to the ischemic side of the brain, which is the left side in this case. And we have shown that the accumulation of the liposomes happens in a biphasic manner, where we have the highest accumulation when injected half an hour after the stroke and also 48 hours after the stroke. So this accumulation was very specific to the lesion area, and it's also co-localized with the area of the disruption of a blood-brain barrier damage. So these data were very promising, and therefore we hypothesized that we can achieve similar selective targeting in ICH with the loss of the blood-brain barrier integrity being the primary mechanism by which the entrance of the nanoparticles will be permitted into the brain. So to prove our hypothesis, we have induced the intracerebral hemorrhage in mice by using the collagenase model. So for this, we injected the bacterial collagenase into the striatum of the brain. And this enzyme will degrade the extracellular matrix basement membrane, and this will allow the leakage of the blood product into the brain. After induction of the ICH, we then injected liposomes intravenously. So we've used either fluorescently labeled liposomes or radio-labeled liposomes in order to be able to follow their accumulation into the brain. And we injected those liposomes at different time points after ICH, 3 hours, 24 hours, and 48 hours, in order to cover the different stages of a blood-brain barrier disruption. After that, we've used either in vivo real-time imaging with SPECT-CT, MRI, and optical imaging, and also we validate that with ex vivo histology in order to analyze the accumulation of the liposomes into the brain and correlate that with the changes in the blood-brain barrier integrity and also with the accumulation of the ion accumulation within the brain, which is an important factor in the blood-brain barrier disruption. So we'll start with you with the SPECT-CT imaging data. So in these images, we are looking at the accumulation of the liposomes within the first hour after injection. So as I mentioned, we have 3 time points of injection of the liposomes, and each time point represents a separate group. So what we could observe is that within this first hour of imaging after intravenous administration, we could see that there is a selective accumulation of the liposomes into the hemorrhagic region of the brain that is observed when the liposomes injected acutely after the hemorrhage, so within 3 to 4 hours after inducing the hemorrhage within the brain. We couldn't see much accumulation when the liposomes were injected 24 hours or 48 hours after the hemorrhage. And at the same time, we couldn't see any accumulation of the liposomes in the healthy side of the brain, which is the left side. And also, we didn't see any accumulation in healthy animals. So this is indicated that this early accumulation of the liposomes in the acute stage is probably driven by the active bleeding process. We then re-imaged the same mice 24 hours after injection of those liposomes, and we could see that the signal in that case, it's much higher and when the liposomes were injected in the acute phase, which indicated that there is a continuous accumulation of the liposomes into the brain over time. Again, this was very selective to the lesion side. We could also observe some accumulation of the liposomes from the 24-hour group, and again we could see another phase of liposome accumulation when injected 48 hours after the hemorrhage. Similar to our previous images, we didn't see any selective accumulation from the healthy animal. So when we quantified these signal within the brain, we could see that there was a significant biphasic accumulation of the liposomes within the brain. So the liposomes tend to accumulate into the hemorrhage side when injected acutely 3 hours after the hemorrhage, and also 48 hours later. On the other hand, when we looked at the clearance of the liposomes into the cerebrospinal fluid, we didn't see much difference between the groups, which indicated that once the liposomes are getting into the brain, there is a very limited clearance afterward, and this has explained the enhancement in the signal that we observed over time. So to validate these data, we also used optical imaging coupled with the injection of fluorescently labeled liposomes. So these images are taken 2 hours after intravenous administration of fluorescently labeled liposomes post-ICH. And again, we see that early on after liposomal administration, the highest accumulation was observed in the acute phase. And when we look at the distribution of the liposomes within the brain, we could see that at this early stage of liposomal administration, the liposomes covering more or less the whole region side, and this is very selective to the ipsilateral of the side of the brain. We didn't see much accumulation from the healthy side of the brain, and similarly, we didn't see any accumulation in healthy animal. When we look at the accumulation of the liposomes that are injected 24 hours and 48 hours after hemorrhage, we could see that this was very much limited to the periphery of the lesion, and we didn't see any accumulation within the center. And this might be explained by the absence of the active bleeding at this time point, and also due to the clot stabilization that is happening at this time point after the disease. So then we followed the accumulation of these fluorescently labeled liposomes 24 hours after the injection, and our optical imaging data again confirmed that there is a biphasic accumulation of the liposomes happening both in the acute and the subacute stage after ICH. The distribution of the liposomes within the brain again was covering the whole lesion side when injected in the acute phase. No much difference was noticed from the accumulation of the liposomes that are injected 24 hours after the lesion. And what was interesting is that the distribution of the liposomes that are injected 48 hours after the stroke has dramatically changed. So we can now see the liposomes almost covering the whole lesion area, and you can see them both in the core and in the periphery. So this is suggested that the initiation of the hematoma resolution and the lysis of the red blood cells that is happening at this time point might contribute to this change in the distribution of the liposomes into the brain, and resulted in this dramatic change that we observe here. I would also like to mention here that in all these time points, we see that the distribution of the liposomes or the localization within the brain is very much co-localized with the region of blood-brain barrier disruption, which is measured by the infiltration of IgG into the brain. So endogenous IgG under normal circumstances, they don't have the access into the brain because of the intact blood-brain barrier. However, when this integrity is compromised, then this endogenous immunoglobulin get the access into these region within the brain. So this is a technique that is usually used as a biomarker for blood-brain barrier disruption, which is good enough to validate if the blood-brain barrier disruption is there or not. However, you can see that from these images, we can't really identify the exact timing of the blood-brain barrier disruption. Therefore, it was essential in our study to validate this timing of a blood-brain barrier disruption with another technique. So For this, we have used dynamic contrast enhanced MRI that is used to measure the trans-blood-brain barrier leakage rate of gadolinium DOTA. So gadolinium DOTA, this is an intravascular contrast agent that in normal circumstances doesn't have access into the brain. However, it gets entry into the brain when there is disruption to the blood-brain barrier integrity. So looking at the gadolinium DOTA leakage index, we could see again that there is a biphasic increase in the gadolinium DOTA entrance into the brain, which is happening both at 3 hours and in the subacute phase. And also the spatial and the temporal distribution of gadolinium DOTA into the brain is very much identical to what we observed from the liposomes. So this similarity in the distribution and the timing of the penetration of liposomes and gadolinium into the brain suggested that the entrance of both of these molecules is driven by similar mechanism, which is due to the disruption of the blood-brain barrier. So an important thing to mention here is that within ICH, the accumulation of parenchymal blood components into the brain, such as fibrin and thrombin, has an important role in the blood-brain barrier disruption. And therefore, it was essential also in our study to quantify the iron content within the brain. So for this, we looked at the accumulation of iron with the T2 star signal. So we could see here that there was a sharp reduction in T2 star signal 3 hours, and this is an indication of the accumulation of iron-filled red blood cells within the brain. And you could see that this was also covering the whole lesion site at the early stage. However, later on, this was mainly restricted toward the periphery of the lesion. So in order to understand how this is correlate with the red blood cell lysis, we also looked at the accumulation of the free iron into the brain with a Prussian blue staining. So we could see that the free iron accumulation within the brain increased over time, and this increase was both in the lesion core and in the periphery, which suggested that the lysis of the red blood cells correlate with this hypointense region of iron accumulation and explain these changes in the distribution of the liposomes over time. One more thing, we looked at the uptake of the liposomes by different cells, and we could see that there is a significant accumulation of the liposomes within the microglia, but we didn't see much accumulation in the astrocyte. And this difference in the accumulation is probably related to the high phagocytic capacity of the microglia, because they have an important role in the clearance of red blood cells and cellular debris. So with this, we have shown you in this study that we can provide an evidence that the liposomes can selectively accumulate within the lesion site in a collagenase model of ICH. Liposomes entry peaked both at 3 hours and 48 hours after ICH, and this is correlate also with the changes in gadolinium DOTA accumulation, which suggested that this is associated with the changes in the blood-brain barrier permeability. So these observations are quite exciting, and it's provide a promising technology for ICH treatment, where we can enable time-specific new therapies, and we can also repurpose some of the drugs that have been tested before in ICH but failed because of the limited penetration into the brain or unwanted peripheral toxicity. So with this, I would like to thank all the people who have been involved in this study, and also thank you all for listening.
Thank you very much for your presentation. We are a bit late, so we will keep the question for the end of the session. So I think we can move to the next presentation by Dr. Camille Page from Research and Development Department in Innomedica Holding in Bern. The title is Talinoren, a regenerative nano drug against neurodegeneration.
Hi everyone, welcome. Thank you very much for the nice introduction, Mr. Berger. Here, I would like, and I'm really happy to present you Tyrannostic. I have been working 3.5 years on that project, on that product. And here you see as a first image, that was the first trial fillings, actually from the first scale up. So it's from R&D to GMP production. We have been working very hard to acquire preclinical data, and at the same time making sure that the translation and the translatability of the product into a GMP product can be achieved. And the aim of a clinical batch is very imminent. So we are planning our first clinical batch production by beginning of next year. So I would like, oh, wait a minute. Yes, okay, sorry. So the overview of my presentation today will be present you Tyrannostic, what does it stand for, the API of the product, GM1 ganglioside, the relevance for Parkinson's disease, because it's where we started, that was our first anchor point, followed by the distribution showing the potential of the product, and where we have been looking and checking for it. So also in other diseases such as amyotrophic lateral sclerosis, we think we can open doors with this product. And I will finalize with conclusions and acknowledgements. Thank you. So Tyrannostic, where does the word comes from? It's for targeted liposomal. So we have a liposome. You can see a model of Tyrannostic. It's a unilamellar membrane bilayer, which is cholesterol rich. So it could be raft-like. So it's a liposomal formulation that has a high content of cholesterol, allowing the accumulation of certain compounds and promoting the take-up by the cells in a very efficient way, actually. And it's composed of only body-owned fatty acids. So all the toxicological parts based on chemical products that are unknown or to the body are excluded to that amount here. And neuroregeneration is for the API, and its most impactful characteristic. GM1 gangliosides has been published in many ways. We have all heard about it. It's everywhere and anywhere in many different pathways in correlation with the central nervous system and even more. So neurodegeneration is maybe one of the biggest hope that GM1 has as a property, right? So GM1 is a glycosphingolipid, and it has been actually documented, as mentioned, to be involved in many pathways. So it's tightly regulating the calcium homeostasis. It's involved in the tyrosine kinase processes and nerve growth factors. It has also been actually documented to be essential for calcium-dependent neurotransmitter fusion at the synapses. So overall, it's neurotrophic, it's neuroprotective, it may be, and it has been documented to be neuroregenerative, and it also has been documented to reduce stress, cellular stress, such as mitochondrial stress and AR stress. So what happens if it's depleted? So if you don't have any GM1, one of the diseases that has been actually shown to lack not only dopamine, we always talk about the dopamine in Parkinson's disease, but it's actually a systemic reduction of GM1. It's in Parkinson's disease. And the clinical proof that was conducted in the Western countries was, the last study was actually conducted by Jay Schneider in 2013, where they could show there that the treatment of GM1 is actually promoting the it's promoting actually to be like, has the potential to be disease modifying. So with this, we thought, okay, let's go back to our liposomal formulation. We have the reason why the clinical trial didn't go further were because of marketability, but also because of the side effects, right? So GM1 ganglioside is like a soap, and when it's injected in a subcutaneous intramuscular way, as it has previously been done, these are terrible pain for the patient. So for a chronic disease like Parkinson's disease, this is not bearable. And we thought, well, with our liposomal formulation, we may be able to actually open doors to a different route of administration. In China, for example, they administer free GM1 by intravenous ways, which are a bit, it's also not so practical for chronic disease. Let's put it that way, if you have to administer every day or every week. So we tested actually, and we managed to have significant dopamine rescue levels in the mouse MPTP model of Parkinson's disease, where we treated actually the GM1 once a day for 14 days upon the trigger of the model by the typical 20 milligram per kilogram MPTP subcute. And we could achieve, actually, if you can see the two graphs, we could achieve that if we compare the free injected GM1 intraperitoneal, which is the equivalent of the subcute and intramuscular administration conducted by J. Schneider in the clinical trials, we could rescue in the mouse model with our Tyrannostic intraoral administration, actually the same kind of levels in terms of dopamine in the striatum. So this opens actually new doors and probably an oral administration for chronic diseases. So to that, we have been working very hard to conduct these results, and we will continue this. But when we analyze the biodistribution, we find some puzzling things on that aspect. So here I show you the biodistribution in a qualitative way. So we did some whole body scan, basically with a near-infrared targeted Tyrannostic and other types of liposomes, which actually are differing in their membrane lipid composition. And we can see that we have an increased fluorescence signal in the brain when you have the accumulation, like compared to the other products after 24 hours after the administration for n = 3 mice. So this was an IV. So we did IV, per os, intranasal. We did many different ways to administer. And this is a per os administration of Tyrannostic in red. It has been labeled with DII, actually. And the dopaminergic neurons are marked in green with the tyrosine hydroxylase, like usual. And you can clearly see that we have some accumulation after the per os accumulation administration in mice. Now, the puzzling things actually is that these are qualitative. So you don't see the liposome itself, you don't see the GM1 itself, but we see the dye and how it could be targeted and accumulated in the brain. When we did actually a quantitative analysis on that, and you see on that graph the free GM1 IV versus Tyrannostic per os and Tyrannostic IV. And we clearly see that by intravenous accumulation, we have almost a doubling of the amount of GM1 accumulating in the CNS compared to the free GM1 IV. Whereas by the perioral accumulation, we don't see a significant or any difference. And the puzzling thing is like, well, but why did it work then in the mouse MPTP model and in the wild type models? When we do the biodistribution, why do we have color accumulation, a signal accumulation? And in terms of, we are still tackling this because we are now planning on analyzing actually the endogenous versus exogenous GM1 by deuterated GM1 analysis. So this is a big study starting now. But one of the proposed mechanisms is for the cellular uptake. Based on our experience, it has been shown that it actually is taken up by endocytosis and redistributed throughout the membranes. With this, we have been following other diseases and indications. And I'm really happy to show you the data on amyotrophic lateral sclerosis. This has been like the breakthrough of the last few months. Amyotrophic lateral sclerosis is actually a motor neuron disease where actually the connection from the brain to the neurons is diseasing and dying over time. It's a very aggressive disease. The life expectancy is about 3 to 5 years. The dying of the motor neurons can start more like at the bulbar phase is more like starting to lose the speech first. And the spinal type of ALS is more like starting by the extremities, leading to patients getting into wheelchairs pretty early. So it's a rare disease, and there is no useful therapies for that. There is edaravone and Riluzole that were both released in the last years, were in some countries of the world. And there's the survival expectancy upon these treatments is 2 to 6 months. So it's not much, and it's actually accompanied by a high, quite some side effects. Yes. So here again, this touches mainly the spinal cord. So we check the biodistribution of talinurin in the spinal cord. This is a tissue section of talinurin labeled with DII in the ventral horn of the lumbar spinal cord part. And here you see the motor neurons clearly stained. I boost a bit the contrast so to make sure that everybody can see. But these are amazing images. And with that check of, okay, it's accumulating, then we have now the thought, okay, GM1 has the potential actually to strengthen neuromuscular connection at the right place, reduce stress factors at the cellular level, prevent motor neuron degradation, and promote the regeneration of skeletal muscles. So these are the results that were gathered at the Inselspital, the Department of Neurology in Bern. And I will start the two. So you can see the mice, talinurin versus saline, wild type versus ALS model. And you clearly see the ALS mouse that is treated with talinurin is pretty well holding up with the wild type mouse. So this is one movie. If we go now to the results of that study. So we actually did postnatal, we started the treatment with talinurin IV, 15 milligram per kilogram every other day at postnatal day 110. And the study is still ongoing. And we also conducted a hanging wire test in addition to the Rotarod protocol. For the Rotarod protocol, you can clearly see the treatment start, and then you have the slowly increase actually over 10 days of the motor performance of the mice. So you have in pink the ALS model saline, in red the ALS model plus talinurin every other day, starting from postnatal day 110. And you have in green and black both wild type, ones with talinurin, the green one, and the other control is the wild type saline. And you clearly see a highly significant motor capacity, motor performance rescue that is actually maintained over time. So when we go to the hanging wire test, where the mice are hung to the cage, and the average time to the first fall and the average of amount of falls are counted. You can also clearly see that for the amount of animals that were conducted in this first pilot, there is not so many animals, but you can clearly see that you have an interesting way, like the average of falls, you have almost like very little for the wild-type talinurin and the wild-type saline with the ALS talinurin are almost equivalent, which is actually, I think, very interesting. So in the overall, we could say talinurin, it shows significant rescue of the motor performance. It has a high potential for being disease modifying. And there is clearly a marked therapeutic potential for talinurin in this orphan disease. So in conclusion, that talinurin is essentially composed of body-owned molecules, which make it very well tolerated in all the animal models we were testing. It reaches the CNS. It has the potential to have a therapeutic effect by oral administration for Parkinson's disease and chronic diseases where the disease is lasting over 10 years. It positively rescues dopamine levels in the stratum of the MPTP mouse model of Parkinson. And it significantly rescues the motor impairment in ALS mouse model. It also has the potential to be therapeutically relevant for several neurological disorders, especially in the fields of neurological diseases. Okay, so in the overall, we could say talinurin is opening doors. It's opening doors by the property that GM1 has and its involvement in different pathways. But in addition to that, the carrier itself, without the GM1, allows us to imagine many ways of treatments for neurological diseases. And maybe to finalize here, I want to show you this was our first technical batch that was actually produced about exactly 1 year ago. It's going to be the 14th of December. It's exactly 1 year that we have produced of first clinical batch. I would like to thank the Inomedica team. We are a small but still growing team. We are all very motivated to bring talinurin to the patient. I would like to thank collaborators and partners that for that specific product are still remaining a bit silent, but that's going to change in the future. And I thank you for your attention. And if you have any questions.
Thank you very much. Only one question? We're a bit late.
Yes. Sorry.
Nobody? Any questions? So we will discuss together at the end, and we can move to the last presentation from Dr. Julie Wen-Wang, Senior Research and Teaching Fellow in Nanomedicine at the Institute of Pharmaceutical Science at King's College in London. And the title is Developing Polymeric Nano-formulation for Neurodegenerative Disease via Intranasal Delivery.
Hello, can you hear me and see my slides? I guess it's on, right? From François.
Okay, you can speak. We see the slides. Perfect.
Okay. Yeah, so good afternoon, everyone from London. So I'll just go to my slides straight away. So as we all know that drug delivery to the brain is never an easy task. So when the drugs are administered systemically, there are multiple limitations. There's liver first pass effect, and then the consequence metabolism and enzyme degradation. And there's also the presence of the BBB and also concerns of the peripheral side effects. With this regards, nose to brain delivery present an alternative route to deliver the drug in a unique way. It's a direct delivery through the olfactory and trigeminal nerves pathways directly to the brain. So the technique itself is non-invasive, and the nasal cavity has a huge adsorption surface area, and apparently there is no liver pass metabolic the effect, and this delivery route also can bypass the BBB. However, there are also disadvantages of this delivery route. The administration dose for nasal delivery is relatively small compared to other routes. The maximum volume for human is around 200 microliter. And then there is the presence of the mucus, which will have where there is clearance and the enzyme degradation happening in the nasal cavity. And then the dosage form, if it's not so muco-adhesive, there will be a possibility to drain down to the GI tract or the lung, and then the retention time is not long enough to enable sufficient drug to deposit to the cavity. So looking at these issues, we thought about that nanomedicine and nanoformulations might provide solutions to overcome these problems. For example, they might be able to increase the drug loading and facilitate the translocation or diffusion of the drug through the mucus, and with modification of the formulation, they might be able to enhance the adhesion to the cavity and then increase the retention time. So our group has been interested in formulation, doing nanoformulations for intranasal delivery. And one of the target we are focusing on is amyotrophic lateral sclerosis, which has been described by several speakers in the clinical events. So ALS is a fatal, progressive, motor neuron degenerative disease with loss of both upper and lower motor neurons. It's still unknown how and then why these motor neurons die, and that's the reason making ALS so hard to treat. So the patients usually have 2 to 5 years survival after the symptom onset, and then patient usually die due to the respiratory failure. It's currently incurable, and despite the fact that there are 2 drugs approved as a disease-modifying interventions. So riluzole being the first drug approved for ALS in the States in 1995. It is a drug to block the glutamate neuron-associated neurotransmission. Patients gain roughly 3 months survival benefit, however, also suffered severe hepatic and then GI side effects. After 2 decades, edaravone was the second drug license for ALS in Japan, and in 2015, and then now it's also approved in a few countries, but it's not approved in EU yet. Edaravone is a free radical scavenger, and it can reduce the oxidative stress inside the brain. The problem with edaravone is very unstable, and therefore the viability is relatively poor, and the patients are required to go to the hospital to receive the slow IV infusion. So our group has been working on nanoformulation of both drugs and for targeting ALS. And today I'm going to share you the work that we try to improve the brain access of edaravone and then reduce its systemic absorption using nanoformulation and nasal delivery strategies. The work I'm presenting today is primarily performed by Dr. Yuan Lu, who was a visiting scientist from Guiyang University in China. So we formulate edaravone in pegylated PLGA nanoparticles. We assess the activity in vitro and then study the biodistribution, quantify the brain uptake by LC-MS. So the nanoparticles were formulated using the nanoprecipitation method. The organic phase contains the POGA-PG polymer, the surfactant, and then the drug. Aqueous phase also contains the trin 80 as an extra additional surfactant to further stabilize the formulation. After solvent evaporation, we obtain the clear suspension of the edaravone nanoparticles with the size around 90 nanometer. The drug encapsulation efficiency is around 20% determined by HPLC. We look at the drug release profiles, and in contrast to the free drug, which shows a burst release within the first few hours, the nano-formulation of edaravone showed a sustained release over time. And up to 24 hours, the nano-formulation of edaravone has around 70% drug release. To test the antioxidant activity, we use the BV2 mouse macroglia cells as an in vitro model. We treat the cell, pre-treat the cells with either edaravone alone or the nano-formulation for 48 hours, and followed by the induction of the oxidative stress with hydrogen peroxide, and then perform the DCFA cellular RS assay. So the DCFA FDA was non-fluorescent. When it was reacted with the reactive oxygen species inside the cells, it would be oxidized to DCF, which is fluorescence, and then we can measure the fluorescence intensity by flow cytometry. So the cells treated with hydrogen peroxide has a high level of ROS, and therefore show the increased fluorescence intensity compared to control. And then in the cells treated with either edaravone or the edaravone nano-formulation, a significant reduction in fluorescence was measured, as you can see here. And the nano-formulation has further reduction compared to the free edaravone. So we move this to in vivo investigation. So we label the nanoparticles with a near-infrared fluorescence dye-R. So we give the mice dye-R labeled nanoparticles nasally or intravenously, and monitor the distribution of up to 4 hours. So from the in vivo, for the whole body imaging, we can see that the signals are limited to the brain area in the mouse treated with the particles intranasally. In contrast, being pegylated nanoparticles, the signals were observed all over the body. The ex vivo image also shows correlate to the whole body image, as we can see that the nanoparticles were taken up in various major organs. And in the treatment group, we see that the signals are limited in the brain. We also observe signals in the GI system, and then this is probably due to some drugs still drained down to the nasopharynx and then under the GI roots. So optical image provide information for us to check the distribution of the carrier, and then how about the amount of the drug remain in the tissues. So further, we perform the LCMS study to quantify the amount of drug after intranasal administration. So we compare IN and then IV injection of the edaravone nano formulation, and then also have the edaravone, free edaravone IV injection as a control, mimicking the clinical settings. The tissues were sampled at 30 minutes, 12 hours, and then 24 hours. And quantification of the edaravone has been challenging. As I mentioned earlier, that edaravone is extremely unstable in the biological fluid and or biological matrix. So initially, we barely can detect Idaveron in our tissue samples. Within days, even the samples were kept in the freezer. And we adapted some methods published from the literature and then also the drug company. So we include the stabilizer sodium disulfide in processing the tissues. And then these enable us to detect the Idaveron by LCMS. And we use ultra performance LCMS, which can determine the drug concentration to picogram per ml concentration. So here I show you the results of the brain and then liver, for example. And bear in mind that due to the injection dose limitation, so the intranasal dose is less than 1/3 of the intravenous injection. However, the comparable up amount of Idaveron was detected in the brain for all the methods and the routes we are doing here. So the IV injection of the free Idaveron, the Idaveron can be detected at 30 minutes but eliminated quickly over time. Nano-formulation has slight retention at 12 hours, and in comparison, the intranasal delivery of Idaveron nano-formulation will have further retention of Idaveron being able to detect in the brain up to even 24 hours. And if we look at the drug in the periphery, so for example, in the liver, as expected, we barely can detect Idaveron in the liver in the intranasal delivery routes. And free IV injection of a free Idaveron, again, the liver can be detected in the liver in early time point, but not later on. And the same situation was observed in the systemic injection of the nano-formulation. So our results, in vivo results, shows that the nasal delivery of nano-formulation of Idaveron outperform the systemic injection of either free Idaveron or the nasal formulation. So in conclusion, we formulate the Idaveron in a nano formulation which can protect it and going through the protect it from the nasal cavity, and then can reach the brain and be quantified by LCMS. And we are very keen to moving on to the therapy efficacy studies in the ALS models. And the model we are interested in is the TDP43 knock-in mice, which has been established by our colleague, Dr. Jamie Sheridan at the King's College London. So overall, the results are quite promising, and then shows that the nasal delivery has a great potential as an alternative delivery route, resulting in a drug PK profiles that may be more favorable for ALS or other CNS disease. And the nasal delivery, as mentioned, that the patients at the later stage, they might even have difficulty in swallowing. So if nasal delivery can be a method, delivery route that are good for the patient, that would be good, more convenient for the patient. So I would like to thank the colleagues, Professor Eljama Suki-Bansal at the King's College London, and then Dr. Yuan Lu and Professor Wang from the Guizhou Medical University. And then Yuan was supported by the China Scholarship Council and spent 1 year visiting our institution and perform this study. And thank you very much. I will be happy to take more questions.
Thank you very much for your presentation. Any specific question on this presentation? No, I just have one. We know that there is neuronal degeneration in the nasal area, for example, in neurodegenerative disease. So did anyone check the functionality of the nasal delivery, for example, in Alzheimer's disease or ALS? Do we have data on that?
You mean the functionality of the nasal delivery for other CNS disease, or
in patients with neurodegenerative disease? Because we know we can make the diagnosis of neurodegenerative disease by cytoskeletal abnormality. So maybe there is an impairment of this, of the functionality of this in this patient.
Oh, I see. Okay, I'm not sure if others have, but I haven't come across that the patient, they might have some functionality changes in the nasal cavity compared to the healthy patient. Of course, the physiology conditions in the patient with motor neuron disease or as CNS disorders might be different. For example, in ALS, some patients it's been reported that there will be a regulated level of the PGP, the drug resistant mechanism. So that also makes the drug delivery more difficult, especially if it's through the systemic injection.
Okay, so thank you. So I think we can move to the general discussion. Again, thank you very much for all the speakers. It was very impressive because we have now tools, overview to explore, to make the data mining, precision medicine, and to cure most of the neurodegenerative and brain disease. So just to open the discussion in a provocative way, and I think this is very important at the COVID time. What is your strategy? Because we all demonstrate there is 2 or 3 presentations that deal directly with human patients, but in nanomedicine, and in the case of most researchers in the world, and especially in the brain disease, we validate with preclinical model, I cure glioblastoma in mice, you cure ALS, Parkinson's disease, Alzheimer's disease, and at the end, we do classical, we have pharmaceutical startup or company here. We do classical clinical trial with phase 0, 1, 2, and 3. And each time we are disappointed. So my provocative question is, do you have an innovative strategy not to be disappointing? So what is your strategy to translate at the bedside with success? Could you, sorry for the questions, and maybe strategy with the innovative drug development you presented, and maybe you can use some of the tools presented by Mira. So what is your strategy? Is it still a classical pharmaceutical strategy, or
I think maybe I can share my opinion on this. I think it's quite critical to understand the pathology of the disease, because probably it's not like one drug that can fit all the patients. Like from the data that I've shown, even with the same disease, the pathology change over time, and probably this is also will change after the initiation of the treatment. So it's important that we understand personally and also progressively how the disease change over time. And I guess generating this large set of data will clearly help to facilitate the translation of the nanotechnologies that we develop.
Okay, thank you.
If I may add, can I, François?
Sorry? Yeah, yes,
can I add something? So I just want to follow up on what you said right now. And I say that if we can share the data among the diseases, even though it's not relevant, we will find a lot of same parameters and they are changing among the diseases. But even more, regarding to your question, François, if we can share even the clinical trials, the failures and the success, that could be the right thing. But you know, we are still very close. We don't share enough. We are afraid. And to say what I said in the beginning, we don't have enough data. We are speaking, we have those phrases, big data, but all what I'm talking always is small data, because we don't have enough really mathematically or statistically to do it the right, reliable way. So I think we have, given the pandemic and given there is no borders and all these threats, I think we should open and share more and find a way to keep our, royalties and so on, but we have to share more, and especially the failures. Okay. Because you never heard if the clinical trials failed. You hear some of the success, and we have wonderful examples from the last years on those who failed or went to market and failed afterwards, which is even worse.
Okay, this is a very important message, sharing and open source. Yes, go.
No, I didn't know. Yes, thank you. I think it's really great, Myra. I think if we could share more, the thing is always how to do it, right? And what is the right form to do it and what would be the platform. But I think in the last years, what I realized actually at Innomedica is that the company built for a tech platform. So we are specialists in liposomal formulation. So in the lab, we produce liposomes and all the according physical chemistry for the analytics. But all the collaborations for all the in vitro assays, it's collaborations with companies, startups, and university labs. And sometimes we are confronted with, but you don't say anything because it's mine, it's ours, I don't want to share. And I always try to say, I think there is enough work for everyone. And then we finally managed to have like collaborations with 3-4 different institutions that maybe wouldn't collaborate otherwise, just by saying, look, the aim is the patients, and every person, every lab, every group has a part of the puzzle, and really to try to respect and find the know-how of each department or each group and put it together. I think this is the combination is the solution. It's like for therapy on the long term. I don't think there is for one disease one solution. Also, Zahara, as you mentioned, like the development of the disease over time. But I think in addition to that, it's also about the complexity and the biochemistry of diseases, right? So I think if we combine know-hows, and we have more chances, actually. No.
And just to add one sentence to what you said, if we can secure nowadays, we have such good tools developed for cybersecurity and sharing of data and keeping so we can develop nowadays technologies to share and to keep you added value and to keep your royalties and maybe be a little bit more generous and say, okay, we want to, as I said, towards society, and we will find a way that everybody will benefit once society is beneficial. Maybe I'm naive, but that's what I think.
Yeah, what you said is very important, because in France and in several countries, we have a huge debate, because the French government create what we name the Health Data Hub. And in fact, in France, we don't have tools. We just have, it was a big scandal because the Health Data Hub was managed by Microsoft. So my feeling is that we have many GAFAM, and so it would be very important that group like you could share the open source tool that we can implement very early, because most often we don't know how the data management, and to have open source tool would be very important, not to be dependent from a big informatic group that is also a problem for privacy. For example, in COVID, in France, I don't know in your country, but all the tracking strategy were violently rejected by the French citizens. So to have something open source, managing privacy, you receive one question about that would be very, very important. You think it's possible, Mira?
I think it's possible. I can tell you more than that the world is changing. And I can quote big companies like IBM and Intel that are now providing and supporting. For example, I mentioned the MIP, and they are offering, I don't know if it will work out, open source. And they found a strategic way to gain from another things, even though it's open source. And I think that has to be, we have to change the regime, and we can do it. And I think it's time to do it. It's not simple because, you know, the word politics, so we have to breach the politics, and then I think we can be better off.
Any more comment? Any more question?
Well, maybe back to your initial question, François. The point about the translability of the animals. So sometimes it's really a nightmare. Already between two studies, if the studies are done on different countries or different labs, then it comes out, we make sure it's exactly the same protocol, you have different results. And for example, for Tyrannostic, I think we decided for actually another type of strategy. We chose GM1 because it was already in human, and it was already positively effective in many type of diseases like Parkinson's, but also for spinal cord injuries and stroke, for example. These are very well documented studies in the Western countries, but also in China. So in China, it's still actively actually administered. So we have now a colleague translating papers. It's actually interesting what they are doing there. And it's really, really interesting. So it's potential. So we put the two pieces of the puzzle together, and then we work with biodistribution. So one of the things is like efficacy, but the other thing is like, does it goes at the target tissues that we want to have, that we want that it acts, right? And with this, you can kind of construct a case based on minimum preclinical data in terms of efficacy, robust biodistribution, and then take the rationale of the risk management based on the fact that the API already has been effective in clinics, right? And that's like the risk is like more like separated, and then you know the day we make the step in the clinic, hopefully next year, then we can expand for other types of diseases because in the meanwhile we will check the biochemistry of the other types of diseases we are looking at. So it's how we try to do it now for Tyrannostic, but it's true, it's difficult. Sometimes we say, well, these models are all not so good, so because we cannot translate, so it's tough. But I think it's getting better. I mean, if you think about it, neurological diseases models, they are like very young compared to, for example, for cancer models. And for example, the cancer models, they are like currently pretty well robust in a way. Huh? So I think maybe it's a matter of time as well.
But
hello, do you hear me?
Yes. Yeah. Go.
Well, actually, this conversation is explaining our approach when we wanted to do a prevention for neurodegeneration. We thought about a compound that will be safe and can be put in the market as a food supplement. And in the meantime, we can do all the animal experiments and gather data from humans, at least for safety and for some from the fact that people can take it without we having first to do the safety trials on it by itself. And we kind of saved many years of work that you all have to do first before you can even approach a human trial. So I think it's very important to remember that in the next future, we won't have the resources to sell people very expensive treatments because they are expensive because you spent so many years developing them. And you have to think about it, life is short. Can I really spend 10 years before I can even go to phase 2 or clinical trial, doing all the before experiments? I have to do something faster and I have to do it cheaper so we can sell it to people eventually.
Any more comments? Maybe just to finish that one opportunity to accelerate is maybe to do more and more fast zero cognitive trial, because with your innovative strategy, you have a target. And now for brain disease, we have molecular imaging, we can do biopsy, we can do imprint. So maybe to check in one patient, is your target reached in the human brain? Do you modulate the molecular and cellular pathway before moving to phase 2, 3 study? So this is not expensive. We have to be very careful with ethics because it's quite like N=1 patient cognitive study, but I think it's probably an opportunity.
At the end, you'll have to see if it works on human.
Yeah.
You can do all the markers that you want, but unless you have a clinical trial that shows works or doesn't work, it doesn't matter. Everything you have done before can be thrown to the garbage if it doesn't work on humans.
Thank you very much for.
maybe there is.
Sorry to say it like this, but I mean, we all know this is true.
Maybe there is another point. Actually, we think always about animal trials, but there is another aspect. Actually, I think it's when you are early in development, the research. Our scientists, the first thing they think about is, okay, I have an idea. Great. So they do their idea. We check our idea, but can we translate it in terms of raw material, supply chain? Because I've seen so many academic projects that are amazing, super great, but you just cannot buy the stuff in big amounts for clinical trials either. So
I totally agree with you. You have to think about
keep in mind
the last experiment, and go as fast as you can, at least to an animal model, and see if your idea is feasible. Before you do the cell experiments and all these mechanistic experiments and all these things, you have to show at least in an animal model that it works and you can make it. Because if you cannot, then it's very nice to get a professor out of it, but you won't have a company and sell it.
Okay, so I think it's time we should discuss all the day. I think I want to thank again all the speaker for the presentation, for the discussion. I would like to thank again Bit Loeffler to organize this meeting. It was very important, and the discussion that was emerging here is really the proof that we need to mix together. So I hope that next year we will be able to come to BAL. So thank you very much.